Hi you guys, welcome to part 4 of muscle. And what we want to get to now is to actin. So we've been talking about myosin. We want to look at what actin looks like. So actin is a little more complex. It's not just one protein, but it's various proteins put together. So let's look at those. It's the orange here. Oops. And as I enlarge, right, there's where my arrow is. And if I were to enlarge that, then I get to this molecule here. And get my face out of the way. And I wanted to show you the details of all this that's going on, because we have the little orange balls. That's the actin. It's called G-actin because it is globular actin. It's in globules or in balls, so that works. But then along with it, there's two other proteins, and one is the troponin, and the other is the tropomyosin, and they're listed here. Tropomyosin then would be the ribbon here, and troponin would be the snail-like structure that's right there. Okay, so this is the entire complex of actin is made up of those three, of the globular actin, of the tropomyosin, and of the troponin. Okay, and they have very distinct function. I want to switch over to full mode here and show you a little bit with my own drawings for you to get an idea of how that works. So if we have those globular protein going on like this, and then you see that it's sort of a double layer, right? It's twisted over itself. And then you have on here this snail-like structure that's the troponin. Okay, so let's write that down, troponin or troponin. And the tropomyosin. So first of all, I'm going to show you that the myosin that are the golf club heads are going to be able to come up and attach onto the troponin and move this. Okay, but in order for it to attach, it's got to actually attach onto a very particular site here, which is going to allow that myosin, the golf club head, to come up and attach onto it like that. Okay, so we want it to be able to do that when there's a contraction. But when there is not a contraction and when the muscle is at rest, you want to be able to block these attachment sites. And that blockage then is where my ribbon comes in. So I'm just going to draw it like this. Not very accurate for all you wonderful artists out there. And if I were to put this ribbon over this, then I would no longer allow for the attachment here because I have blocked that location where the myosin can actually attach. So this muscle is at rest. So this ribbon then is the tropomyosin. Okay, now what has to happen in order for a contraction to occur is you have to move out the tropomyosin. So what is going to trigger that movement of the tropomyosin? And the whole secret lies in calcium. So if I have calcium that trickles down here from the sarcoplasmic reticulum, okay, for now let's just think of it that way. It's being released. How? We'll see that later. So it's being released, and calcium is going to attach onto the snail-like structure, which is the troponin. When it attaches onto that, it changes shape a little bit. Like any molecule that has a new atom attached onto it is going to change shape. It's going to look different. And because it's going to change shape, I'm going to do some troponins down here. So we're going to have these like this. And once those change shape, because they too have calcium here attached onto them, then what they do is they drag the tropomyosin with them out of the way just by moving, just by changing shape. Okay, so once that calcium attaches, notice that up here on my other one there'd also be this ribbon. Okay, what would happen is you would remove the blockage of the attachment site of myosin, and myosin would now be able to attach, and it would be able to attach, engage, and pull. Detach. Come up, attach, engage, and pull, and detach, and keep moving this so that the actin is going to slide. Okay, I'm going to go back to my slides again for a minute. And show you that with much better illustrations. So here's the G. Now here you see the tropomyosin that's blocking the attachment, and it's two different rows of it. This is just written on your notes so that you can go back to it again. And nothing that I haven't told you, so let's move on. This is where the calcium is going to come in now. Okay, so I have calcium coming down and attaching onto troponin, moving tropomyosin out of the way and liberating these attachment sites so that now the myosin head here can go up and attach. Let me get my pen. And it's going to, come up and attach right in that spot there and in each one of these. And drag it, move it along. So that's really the complex mechanism that exists inside a muscle cell. And it's going to allow for this muscle cell that was this long to slide, because the overlap of myosin and actin may have started not very intense, and then it's going to continue to slide, slide, slide. When will it continue to slide? As long as there is calcium, it will continue to slide. So let's continue on here. Here's pictures comparing when tropomyosin blocks that attachment site and when tropomyosin is out of the way of that attachment site, allowing for the contraction to occur. So these diagrams are just showing you that when you want that shortening, what it's going to look like, right? We want it to be extended out like this, where here's the myosin in purple that doesn't move. The actin in the reddish is going to move in this direction. It's going to move in, and it's going to go from being this wide to less wide. And so that muscle is going to bulge up, and that's what a contraction is in your muscle. Okay? So what the next question that comes up right away is, where does that calcium come from? I mean, why is it floating around there all of a sudden? Well, first of all, where is it stored? And that we went through, it's stored in the sarcoplasmic reticulum, and it's all this blue area here. So look, it's absolutely surrounding a sarcomere. It is so close that all you need is a signal for it to actually release that calcium, and you're going to begin a contraction. Okay? The next question is, what is that signal? So just before you go on, I put this in your notes so that you can be sure that you distinguish the difference between troponin and tropomyosin. Be sure you know that. Okay, so what we're thinking of now is, what is going to trigger that release of that calcium for all of this on my board to actually be triggered? Well, it's a neuron, right? It's a neuron that is coming over to the muscle and is going to be sending that signal and saying, okay, go. And then that cascade of events is going to be occurring. Okay? So there we have to integrate. We have to think now of what's happening with the neuron, what's happening with the nervous system that is triggering the muscle to contract. That's going to be for the next video. Let's stop there. Thanks.